Here in the US, 1 person in 10 has diabetes, and while there are treatments available, they're not fun. And for most patients, we don't have a cure. In diabetes, the body loses the ability to regulate blood sugar levels because a particular tissue type, known as the islet, stops working. And if we transplant healthy islet tissue into a diabetic patient, blood sugar levels are normalized and the condition is resolved. However, there's simply not enough donor tissue to go around. So researchers all over the world are developing methods to build islets in the lab. And while they've had a lot of success with sourcing the appropriate cell types, turning those cells into functional tissue has been challenging. Well, if you're trying to build a chair that you can safely sit in, not only do you need to have the right parts, but you must also assemble them properly. Similarly, if you're trying to build a functional tissue, not only do you have to have the right cells, blood vessels, and extracellular proteins, but you must also assemble them properly. In short, if you want proper tissue function, you must also have proper tissue structure. So I hypothesize that the difficulties we're having with building functional islets come not from not having the right cell types, but rather are the result of not turning those cells into the right tissue structure. So if that's the case, it's not surprising. I mean, it's not like we have an instruction manual for how to assemble cells into functional islets. And have you ever tried to build IKEA furniture without an instruction manual? Yeah, pretty much impossible. So I'm writing an instruction manual for how to assemble cells into functional islets. And it has 2 parts. First, I'm drawing a diagram of how the pieces of the tissue should fit together. And I'm doing this based on high-resolution 3D images of healthy islet tissue, in which I am going in and I am measuring areas of contact between adjacent cells and between cells and blood vessels. And there was no way to do this when I started the project, so I had to invent one. Second, I'm learning the step-by-step instructions that cells use themselves naturally during development to assemble themselves into islet tissue. We know that cells will organize themselves in tissues based on inherent cellular characteristics. So I'm going in and I am measuring those characteristics in healthy islet cells so that we can understand the process that the cells use naturally to assemble into functional tissue, and then use that same process when we're assembling cells into functional tissue in the lab. In the end, I'll have an instruction manual that other researchers and I can use to build functional islets that we can implant to cure patients of their diabetes. Thank you.